So today, I mean, as you can see from the title, I will talk about sleep and consciousness, and it's really two concepts, sleep and consciousness, that have shaped my scientific career. My main interest initially was in consciousness. When I was looking for internships during my graduate studies, I had the opportunity to get in touch with Giulio Tononi at the University of Wisconsin, who really told me how to embrace this approach of consciousness, which is looking into sleep. And something very interesting about the relationship with sleep and consciousness that I will try to pass through this presentation is the fact that in recently we have a more nuanced approach of the impact on sleep of consciousness. So often you have this way of even defining consciousness, which is the thing that you lose when you're falling asleep, and that you regain when you start dreaming, for example. And it's very interesting to see in recent research how we are nuancing this vision, and how we can see that there is a much more complex relationship between sleep and consciousness. So it's really what will be the core topic of this talk. And something that has been really present throughout my career. From my early work, so before I started a PhD, I had this opportunity, as I said, to work with Giulio Tononi and Yuval Nir, who was at the time a postdoc in Giulio Tononi's lab, and who has now his own lab in Israel. And the work I conducted there was to very much an electrophysiology work. So looking at the brain activity of humans, so in this case, epileptic patients, while they were either awake or sleeping. And this work was made possible by the fact that these epileptic patients are implanted as part of their clinical care with depth electrode. It gives us opportunity to look at brain activity at pretty much all scales of recording in humans, from the single neuron to the whole scalp. And what we found is that sleep is much more complex and much richer than what is typically presented in textbooks, for example, and especially that there is this local component of sleep. There have been accumulating evidence from the Tononi groups and other that sleep can be seen as a local and locally regulated phenomenon, at least at the level of cortical areas. So after this initial work that was really focused on the electrophysiology of sleep and this concept of local sleep, I moved to a PhD in Paris under the supervision of Sid Quider. The idea here was to look at the functional consequences of local sleep modulation within sleep, and especially whether complex cognitive processes can be supported in the sleeping brain by the fact that you will have modulation of sleep depth. So can, in a way, your brain wake up slightly and locally to allow you to interact with your environment. So that will something I will talk in the first part of the talk, and that constituted my main PhD contribution. After that PhD, which ended in 2016, I moved to Australia. You can see there is a gap between the two, 1 year gap that I took with my wife to travel a bit and think about the next step. And I arrived in Australia then in 2017, working both with Joel Pearson in Sydney and Nao Tsuchiya, whom I'm sure a lot of you will know. And there I decided to do a bit of a break in terms of sleep studies and study wakefulness and the fluctuations of attention. So it's a bit of the the other side of the coin. So if you can understand how consciousness is fluctuating during the night, can you use the same tools and approach to study how it's fluctuating during the day as well? So I was very interested in the fluctuations of attention, not only their impact on behavior, but also their impact on subjective experience, instances of, for example, mind wandering. And what we found there is that we can have the reverse phenomenon as what we have in sleep, which is that local modulation of the vigilant state of the brain. So local sleep induction could explain why sometimes our attention fluctuates and why sometimes we're mind wandering, for example. So to finish a bit my, in a way, self introduction, sorry, just turning up. Yeah, I'm back from Australia since earlier this year. I'm now at the Paris Brain Institute, which is a big neuroscience institute in Paris. And with two other colleagues, we are launching a new team that we have very humbly called the Dream Team. Not necessarily because of the members, although I'm very, very happy to work with Professor Isabelle Arnulf and Dr. Delphine Oudiat, but that because our core topic of investigation will be sleep, dreaming, and consciousness. And we're focusing on this phenomena through both basic and clinical research. So looking at the healthy brain, but also the pathological brain. So this team is in the making, growing up as we speak, and we'll be very, very happy to answer especially students' query if you're interested in the research we're doing here. So back to my own approach of sleep. So as I was mentioning earlier, there has been a shift recently in sleep research from a concept of sleep as seen mostly as a global phenomenon toward something more local. So the classical presentation of sleep is that sleep and wakefulness are mutually exclusive phenomena. So either you're fully asleep or you're fully awake. And when you're asleep, the characteristic of deep sleep is a radical change in neuronal dynamics. So if you can recall, for example, the brain from scalp EEG, when people are deeply asleep, what you will notice is the occurrence of this huge train of slow waves that I've depicted on the screen. We'll try to put the pointer because I don't think you can see what I'm pointing out. Well, never mind. So on the EEG trace, you can see when people are asleep, these high amplitude slow waves that translate a state of hypersynchrony of the brain. At the neuronal level, these slow waves correspond to oscillation between state of activation and inactivation from vast assemblies of neurons. So you have the neurons that are getting active in synchrony and then inactivated. And this oscillation, this bistability of neuronal activity has been thought to explain why we're losing consciousness in deep sleep, because it's radically different pattern of brain activity compared to wakefulness. So in wakefulness, on the contrary, if you recall scalp activity, what you will see is desynchronized oscillations, and at the neuronal level, you will see that neurons have the ability to sustain firing for long periods of time. So very different states in terms of physiological activity. And these states have been thought to be quite homogeneous. Wherever you recalled in the cortex when you're asleep, it was thought that you will see something pretty similar to other brain region. And similarly in wake, if you recalled wake dynamics in one part of the brain, you will see the same everywhere else. Now, we know that there's actually a bit of a complexity missed out by this picture. Recent evidence have suggested that wake and sleep patterns of activity can overlap. Especially during the night, you can sometimes recall brain regions that are recovering patterns of activity that are reminiscent of wakefulness. So some sort of local awakening within sleep. This was first evidence when looking at sleep onset. So one property of the human brain is that it's large. It's a large brain compared to other species. This size might explain why when we fall asleep, the dynamics of the sleep onset period is taking a bit of time. So in this important paper from the sleep group in Lyon in 2010, they showed that if you record individual brain region and you compute an index of sleep through this change of dynamics I was mentioning before, so this transition towards lots of high amplitude slow wave, you can see that this transition between wake activity and sleep activity is not done at the same time for all brain region. There is actually an order of sleep transition within the brain. And this gradual sleep onset takes a bit of time. So in this figure, for example, you can see that between the first brain region to fall asleep, which is the thalamus, to the last one, you have 12 minutes. So a period of 12 minutes in which, if you will take a snapshot of the brain activity within these 12 minutes, you will see that parts of the brain will have already transitioned to sleep and parts will not have already transitioned to sleep. So there is a moment during sleep onset where the brain is caught in intermediary state of a mix of wake and sleep. That could explain some phenomena, such as when you're watching a movie or reading a book while just before sleeping. When you wake up, you might have sometime the impression that you don't really remember the last minutes or the last pages you read. So this could be because maybe you were partially awake when you read or watch this movie or read this book, and the fact that you were only partially awake meant that you were able to read the book, turn the pages, and so on, but you don't remember any of it. These local aspects of sleep is not restricted to the sleep onset periods. It has also been observed in what we call consolidated sleep. So after you have transitioned to stable sleep, within this stable sleep, you can still see regional differences in the way the brain sleeps. So this is an insight that have been made possible by intracranial recordings in humans. So here you have a trace recordings from two periods of sleep. The middle line is a scalp EEG channel, so a frontal to central derivation. And what you can see in this frontal derivation, so the central line, is a classical example of slow wave sleep. So you have a train of high amplitude slow wave developing. So the conclusion, if you had only that information, is that the participant is deeply asleep. This activity is replicated in other brain regions like the DLPFC, but if you look at the motor cortex, something weird is happening. So while the scalp and other brain regions are showing slow wave, what you have in the motor cortex is something opposite. And what you see is a desynchronization, some fast oscillations that are usually observed in wakefulness. So here you have a kind of a paradoxical state of sleep, in which some brain region will show something that's typical of deep sleep. And that's the information you will pick at the scalp level. But you have some other brain region at the same time that are showing something completely different and something closer to an awakening. So even when things, or when people appear to be asleep on the scalp, it doesn't mean that all brain regions are sleeping. And if you have only scalp information, which is most of the time the case when you're recording a healthy participant, you might miss a lot of what's happening deep inside the brain. So this insight and this notion that sleep can be local is something that we have tried to quantify with Giulio Tononi, Chiara, Isaac Fried, and Yuval Nir. So we recorded these patients intracranially, and we distinguish between two situations. So the first one is when sleep is global. So that's what I'm showing you here on the screen. So you can see these slow waves I was mentioning on the scalp, so that the red curve, and below you have recordings from different brain region within the brain. So supplementary motor area, entorhinal cortex, hippocampus, amygdala, and so on. What you can see, the depth EEG is showing something very, very similar to the scalp EEG. The polarity is reversed because it's intranuclear recordings, but you can see that the slow waves that are observed on the scalp are replicated virtually in every single brain region. So you have really here a state of hypersynchrony. And these waves are associated with these alternances of on and off state of activity and inactivation. So that's a very global state of sleep, all brain regions sleeping in the same way. But that's only one way we are sleeping. What you can have also sometimes is these waves occurring in one brain region and not another. So that the second picture I'm showing to you here, where you can see the first wave is both visible at the central scalp electrode, CZ, so that the red curve, and in one brain hemisphere, but not, so the left one, but not the right one. And you can even have situation where you will see a slow wave in the right hemisphere, but nothing on the left and nothing on the scalp. So this is an important distinction. So what you see on the scalp is not necessarily reflective of everything that happens inside the brain. And there are things that are happening inside the brain that you don't see on the scalp. And most importantly, when you quantify how local or global slow waves are, what you can see is that actually the vast majority of waves are local. They are not impacting every single brain region. So the first figure I was showing you is more an exception than the rule. It seems that, at least in the human brain, sleep is much, much more local than was previously thought. So what are the consequences of this local modulation of sleep? If some brain region can, in a way, wake up while we are asleep, does it mean that they can recover some of the cognitive functions that they implement during wakefulness? So that's the question I tried to answer with Sid Quiller during my PhD. And what we wanted to show is whether we could relate local modulation of sleep with the ability to process information while you're asleep and the ability to covertly respond. And by covertly respond, I mean the brain preparing for a response, but not executing in it. So we were not interested in how people respond during their sleep, but with an actual physical response, but whether their brain respond even if they don't move. So to do this, we used a series of paradigms that always follow the same approach. So we had participants come to our lab, so usually healthy sleepers, bit sleep deprived. We were inviting them after lunch, putting them in a dark room, comfortably installed in a reclining chair. And we're asking them to do simple but quite repetitive experiments. For example, in the case I'm showing here, we were asking them to do what's called a semantic discrimination task. So you're presented with words that are belonging to two semantic categories. So in this case, either an object word like knife, table, spoon, or an animal word like sparrow, dog, cat. And participants just had to indicate whether the word they heard was an object or an animal. And they had to do that with lateralized hand response. So for example, click on the left if it's an object and click on the right if it's an animal. Of course, this pattern of response was counterbalanced across participants. So participants were instructed to do this very easy task. The rhythm of the task was slow. As I say, they were comfortably reclined in a dark room. So very often this participant will fall asleep during the task. We'll monitor their brain activity with scalp EEG. And what we will do is to change the list of stimuli as soon as they have transitioned to stable sleep. So from the perspective of participant, the task will be identical. So the stimulus category will be the same and so on. The only thing that will change between wake and sleep is that they will be exposed to new words during sleep. We wanted to expose them to new words during sleep because we wanted to force them to process these words and not to rely on things they would have learned during the wake period after falling asleep. And to be able to see if participants were processing these words while being asleep, what we did was to record an index of motor preparation, which is called the lateralized readiness potential. So in the lateralized readiness potential, what you compute is a difference between the right and left hemisphere according to the expected side of response. Because if I'm about to do a right hand response, that will desynchronize oscillation in the contralateral hemisphere. And the opposite will happen for left hand response. So if you then subtract right and left according to the side of response, what you obtain is a lateralized readiness potential. So here is this readiness potential in wakefulness. It's a negative deflection just before response that is centered over motor areas, and that is supposed to implement all the processes leading to a behavioral response. So what we tried to do here was to check whether this LARP was present when people were hearing these words after falling asleep. And that's the first result we obtained in a first experiment that was conducted in collaboration with Tristan Bekenstein in Cambridge. So we recorded this negativity over motor areas in light non-REM sleep. So a state corresponding more or less to stage 2 sleep for the sleep experts in the audience. It's important to stress that this LRP cannot happen by chance. So if you have no response, no motor response, what you have is no difference between left and right hemisphere and a flat line. So the fact you have a negative deflection, it really occurs because participants are processing the stimulus, associating to the correct category, and associating this category to the correct side of response. So they remember in a way the rule and they implement the rule. The difference we see between this LRP and the wake LRP is mostly a difference in timing. It's much later and much more spread in time than in wakefulness. Interestingly, we tried to replicate this work in using another task. So this was done during a nap. We did a second nap experiment where this time we asked people to do a semantic decision task. So the lexical decision task is you have to decide whether a word exists or not. The semantic one is it's only words that exist. You just have to discriminate semantic categories. What we found there is a similar LRPs. You can notice that the results are a bit noisy because in nap experiment, you don't have so much data, unfortunately. And something quite interesting is the semantic decision LRP was a bit earlier than the lexical decision LRP, which corresponds to the fact that these tasks usually have different reaction times. The semantic decision one is usually faster than the lexical decision one. So even in sleep, we're able to see this difference in the complexity in the way of the task. But in both cases, we were able to see that people were engaging in the task and preparing for motor responses while sleeping. So we tried to see if this result will generalize to a full night. And so we moved from afternoon short naps to a full night recording using a semantic decision task. And we looked again at light non-REM sleep, so roughly corresponding to N2. And what we saw is that we do have index of motor preparation during the night in response to the words that were presented to participants. So sleepers can process words, access to their meaning, and prepare for a response while they sleep. Now, how can they do that? First of all, it's quite interesting to see when this stops. So when they are no longer able to do that. So we look at deeper stages of sleep, like deep non-REM sleep and REM sleep. And we didn't find an LRP in these deeper stages of sleep, which doesn't mean that the words are not processed at all. I mean, you're still encoding them. For example, we're still seeing auditory evoked potential, but we don't see the end stage of the processing chain. So we don't see this LRP anymore. So it seems there are states in which people are sensitive to the external world while they sleep, and states where they are less sensitive to the external world. So how can we explain why sometimes you can respond and sometimes you can't? So we looked a bit more in details in how the brain was responding to the stimuli while people were asleep. And when you present a stimuli in light non-REM sleep, for example, something interesting happens just after stimulus presentation. So that's a time frequency decomposition of the EEG on the top graph of the EEG in response to these sounds. And what you can see is an increase in the power in the delta band, so that the lower yellow blob, and the spindle band, that's the second one, between 14 and 18 hertz. So delta and spindle bands are hallmarks of non-REM sleep. They are sleep markers that are very present when we're asleep, and that have been often associated to sensory disconnection and sleepers isolation. So what happens when you present the stimulus, and the first reaction of your brain is to produce these rhythms, especially in the frontal areas of the brain, as if sleepers were trying to protect themselves from these perturbations, and especially so in over the frontal areas. But then after this initial sleep protective response, what you see is something quite different later on. Is what you see, the same rhythm, so same delta and spindle power is being reduced. And this reduction occurs over central areas, which are auditory and motor areas involved here in the task. So after a first reaction of sleep protection, it seems that there is a shallowing of sleep in the parts of the brain that are engaged in the task, and this reduction in the markers of sleep not only correspond to the LRP in time but also in space. So we are potentially in a situation where when you're asleep, the first reaction of your brain will be to try to not wake up. So to make sure that, especially that the prefrontal cortex is not waking up. But then you could have a local modulations of sleep in other brain areas that could potentially process the external stimuli in isolation and help you decide whether or not it's a good time to wake up or not. So that's could explain why we're able to respond to the stimuli. Now, sometimes you don't, as I said. And especially in REM sleep, we didn't see any LRP, which is quite intriguing, because REM sleep is a state in which brain activity is pretty similar to wakefulness. In French, we call it paradoxical sleep, because you have sleepers in a quite paradoxical state where they, from their brain EEG, they could be awake, but they are deeply asleep. And if you wake them up, they often report dreaming. So if you're conscious, if you're perfectly able to process complex information such as a dream, why are you not able to process easy, simple external information like the words we're presenting to our participants? So our hypothesis here was that it was precisely because they were dreaming that they were not able to process external information. They were, in a way, inattentive to our task because they were too busy and too engaged in doing something else, which is dreaming. So to test this hypothesis about interaction between endogenous information and exogenous information, we try to use a proxy for how rich the experience of someone is. And to do that, we used a metric that was gaining traction at the time, which is EEG complexity and the Lempel-Ziv complexity. So the Lempel-Ziv complexity was used initially by Marcello Massimini's group to detect levels of consciousness. And we tried to replicate this in sleep. And as you can see, if you compute the EEG complexity in different states of sleep, you follow a very expected pattern that complexity is the highest in wake, then it reduces in light and even deep, even more in deep non-REM sleep, and then it goes back a bit in REM sleep. So this is fully follow what we know about consciousness modulation in sleep. Obviously, when you're awake, you're most of the time conscious. Then consciousness tends to fade the more you go toward deep non-REM, and it goes back in REM sleep where you're often dreaming. So we have this metric that seems to track the richness of experience from participants on average. But we wanted to know whether it was able to predict whether or not people will respond to stimuli. So what we did was to compute the EEG complexity just before the sound was presented on the baseline, and to try to see if it was predictive of the ability to respond, so the magnitude of the LRP. And what we found that was a positive correlation in both wake and non-REM sleep. So the more you are, the more the activity of the brain is complex in the mainline, the more you will be able to respond to the stimuli during either wake or non-REM sleep. What was quite interesting is what we observed in non-REM sleep, where we also saw a relationship, but a completely reversed relationship. So in REM sleep, it seems that the more you have a complex EEG activity in the baseline, the less you will be able to process this external stimuli, which was in a way fitting with our hypothesis that the more you're engaged in something internal, the less you will be able to connect to your environment. So it seemed that between non-REM and REM sleep, you have different ways of being disconnected to your environment. In non-REM sleep, you have brain oscillations that can cut you off from the external world. So this slow wave we see in response to the stimulation are quite predictive of your ability or not to process external events. This has been interpreted as a form of cortical gating. So this slow wave prevents information from being distributed across the cortex, and that's why you're losing consciousness and your ability to respond. In REM sleep, you have no or less of this slow wave. So you have to potentially imagine another mechanism explaining why you are not processing external information. And one of the mechanisms we propose is a form of informational gauging. So it's because you're already processing an information, an endogenous information, that you can't, or you have a harder time processing an external one. So far, I presented you work on how people respond covertly to brain stimulation during sleep. So at no point in the data I showed you, people were actually pressing any response button. We were discarding all these results. We're only interested in how the brain responds while the body is irresponsive. But I want to point out some work from my colleagues here in Paris and other groups around the world. We study lucid dreamers. So lucid dreamers are interesting because they are able to signal when they are dreaming with ocular movements or other muscular movements. So they use this ability of lucid dreaming to try to install a dialogue between the experimenter and the sleeper within their dream. So they ask people to indicate when they were lucid dreaming, and then they ask them questions and recording their response using, for example, facial movements. So this is an example of such recording where you have here a narcoleptic patient in REM sleep being asked whether or not they like watching football. And the patient had a signal, a code he could use to answer yes or no to this question. So they were able to ask questions to this participant and record their answer in real time while the person was in a dream. It's very interesting because then when the people wake up, they can actually describe what their dream was. And you can see how there's this weird interaction between the information coming from the inside and the information coming from the outside. And in one of these dreams, the patient actually described how he had to leave the room he was in to go outside, to look at the sky, to be able to perceive the flashes of lights that was presented to them by the experimenter in the real world. So see how try to, in a way, escape or reduce the impact of the endogenous dream information to be able to process external world information. So I think this is beautiful work opening many avenues of research, because it shows how you can even respond overtly to a stimulation while you're sleeping. With Sid Quider, we developed this interest in how sleep is shaping our cognitive abilities in several directions. One thing we did was to look at the ability to orientate attention. So I will speak only briefly about this, but our strategy was to present different auditory streams. So people talking to sleepers. The only thing we did was that one speaker was meaningful, so it was someone speaking in French, and the other was meaningless, so it was a made-up, invented language. So if you don't know anything about French, you wouldn't be able to make to know the difference. But if you know French, obviously you can quickly understand that one is saying something meaningful and the other is saying something meaningless. And what happens is you get attracted to the meaningful stream. Your attention gets captured by the fact that someone is saying something meaningful. So we were interested in understanding whether the same kind of attentional capture occurs during sleep. If I present you the same two streams, one meaningful, one meaningless one, will your brain focus and amplify the meaningful stream? Obviously, here again, we don't have behavioral response. So to measure that, we use a stimulus reconstruction approach. So we're reconstructing the streams that were presented to participants and comparing them. If you're paying attention to one stream versus the other, then we have a better ability to reconstruct that stream. So comparing the level of reconstruction between the two streams allows us to know whether you are paying attention to one versus the other. And that is our results. So the blue is a meaningful speech. And what you can see is that in wakefulness, obviously, people have the tendency to pay attention to the meaningful stream, but that also the case in non-REM and REM sleep. What we could do as well was to dive into details of the electrophysiology and how sleep markers are impacting this ability. And what we saw that slow waves and rapid eye movements are inhibiting your ability to pay attention to a meaningful stream. Blocking mechanism, the same as I outlined before, while other sleep rhythms are actually sort of opening windows of wakefulness within sleep and allowing you to pay attention to the external world. So far, I've talked about the brain activity kind of an ensemble. I wanted to mention this paper from Tamaki and colleagues who studied how you could even have some left-right modulation of sleep depth. So they leverage something that's quite well known in sleep research, which is called a first night effect. So when you sleep in a new environment, usually your sleep is shallower because the environment is new and so on. So what they did was to record slow wave activity in the left and right hemisphere in the default mode network. And what they observed is that when you're sleeping in a new environment, so that day one, your left hemisphere is sleeping less deep than the right hemisphere. And what was very interesting in this research is that they found that this lateralization of brain activity was making people more sensitive to deviant sounds when they're sleeping. So when you're sleeping in a new environment, it seems like you have, in a way, one of your brain hemispheres that's sacrificing for the other, so keeping watch. and being more sensitive to surprising events, to exceptional events, to allow you potentially to wake up if it's needed. And this lateralization is completely disappearing on day 2. So once you get familiar to the environment, you don't need potentially this awake mechanism, this watch mechanism, and then and therefore your true brain hemisphere are just resting as they should. So if you're able like this to process external information during sleep, does it mean that you can learn out of it? So that's something we got very interested on with Sidquidaire. So I will present you a first work we did on lexical categorization. So we were presented words both during wake and sleep, and we asked after the nap to people whether they remembered hearing the words heard during wake and sleep. So classical memory test. We asked them both about an explicit recognition of these words, but also confidence rating, whether they had the feeling that they heard these words in the nap experiment. So there were no sign of explicit memory for words heard during sleep. So as you can see from here, I mean, they really were considered as new items. People were not able to say, yes, I heard these words, even if they heard them dozens of times during the nap. What we did find is a form of implicit memory. So if we looked at the confidence rating, we saw small differences between sleep and new items. So the sleep items were not fully processed or as completely novel items. There were no explicit memory, but in terms of confidence, they had a hunch that some of these items might have been presented during sleep. To confirm this, we looked at the brain responses to these words when they were presented during the memory test, so upon awakening. And we saw that words heard during sleep were processed differently than new items. We had especially early negativity in the scalp EEG response. So their brains remember in a way that these words were presented during wakefulness. But this trace is quite different from the trace we observe with wake items. So they have a memory, but it seems that this memory is a bit different from the memory you would have from wake events. We explored further this notion of learning during sleep using a noise memory paradigm. So it's a paradigm where you try to teach new auditory objects, new sounds to participants. So, and what we use in this paradigm is a paradigm where what we teach to participants is noise. So you present white noise to participants, and in some cases, the noise we present is not repeated, and in some other, there is a repetition within the noise. And what has been shown in wakefulness is when you repeat the same noise snippet several times, people little by little learn from it, and this noise becomes a new sound you can later recognize. So I'm not sure if you hear the stimulus. I don't think I've shared but never mind. I'm running a bit late on time, so I will go on this briefly. To give you a very brief overview of this paradigm is we have this reference noises, so the orange noises that were noise snippets that we presented several times during the experiments. And what you can see with this noise is that little by little participants will learn them and be able to recognize them with a higher accuracy. So what we did was to present some of these references in noise in wake, some other in REM sleep, some other in non-REM, and we tested participants' performance upon awakening. And this is what you get. An index of long-term memory. So you compare the performance for the words that were presented either during wake or during sleep with new items. And what you can see is that there is an advantage of being exposed to these noises in wake and REM sleep. So prior exposure boosts the performance upon awakening, and that's a classical index of learning. You don't see that for non-REM or new items. New items make sense because they're new, so you don't have the time to learn them. What's important to know with this form of learning is that it's very, really fast. So even in the memory test, new items are actually being learned and that are not new anymore. So that's the result we obtain when we focus on the beginning of the memory test. But if you take the entire memory test, then you can see that by the end of the memory test, new items have been learned as well. They are not new anymore. What was very surprising is what happens with the non-REM items. So it's the items that have been heard by the participant by far the more. So they have heard these items hundreds of times during the night, and yet they don't have any advantage for that upon awakening. And if anything, they have a disadvantage because by the end of the memory test, the worst performance obtained for these items heard during non-REM sleep. They are actually even worse than new items. So hearing these noise snippets during the night is detrimental for performance. You're worse off by being exposed to these noise snippets during your sleep than if you never had heard them in your life. So this was a bit puzzling for us because it seemed that we had parts of sleep where learning was possible, like REM sleep, but parts of sleep where we had the opposite effect going on, the form of forgetting. And this forgetting was confirmed by looking at what was happening during the night. So the beautiful thing about this noise paradigm is that we can track the learning with EEG during exposure. And we have an index of how much these items were learned during the night. And what we could see is that at the beginning of the sleep cycle, when sleep is light, we have a form of learning, so that the purple curve here, these items are learned. But the deeper you go into sleep, so the more and more slow wave you have, the more this learning disappears. And it seems that in deeper parts of sleep, these items are being forgotten, as if there was a suppressive effect of slow wave sleep. The suppressive aspect of slow wave sleep was quite interesting because there are theories saying that sleep is actually very important for forgetting. One important difference between wake and sleep, and why we could learn in wakefulness or other states like REM sleep and forgetting sleep, could be because of the changes in the level of acetylcholine. So acetylcholine is a neuromodulator that's very important for synaptic plasticity. And when you have high levels of acetylcholine, you have the application of the Hebbian rule, neurons firing together, wired together. So imagine I present you with a new noise. This noise will trigger some patterns of activation in your auditory cortex. And each time I present this noise, I will strengthen this pattern of activation. So this is what happens when acetylcholine is high. You're reinforcing this trace and improving the memory. Now, what we think happens when we are in deep sleep is since acetylcholine is low, we know that under low level of acetylcholine, the Hebbian rule actually reverses, and neurons firing together actually unwire. So what will happen in deep non-REM sleep is that the initial trace that will be activated by the noise will be erased little by little. So that could be what this mechanism explaining this suppressive effect, that you are actually actively suppressing information when you're asleep. Now, I would like to move to the other part of the talk, which is after seeing what happens during the night and how small fluctuation in sleep depth locally can allow you to recover some wake cognitive function. We'd like to talk about the opposite phenomenon, which is how when you're awake, but you start getting tired, you can have intrusion of sleep activity in your brain locally. So from a global perspective, you're still awake, you're still behaving, but maybe parts of your brain are getting tired, and that could explain why you're missing some information or not paying attention. So this phenomenon has been first evidenced in rodents by a seminal paper from Vlad Vyazovsky, who found that when animals are sleep deprived, you can see sleep-like oscillation in their brain. These sleep oscillations are quite reminiscent of what happened during sleep. They can be global, they can be local, but most importantly, they are associated with a decrease in the firing rate of neurons. So these off states, these episodes of neuronal silencing, which could be detrimental to the processing of information. What Vlad showed is that these sleep-like slow waves increase in frequency the more you stay awake. So the longer you stay awake, the more you will see this sleep-like intrusion. So they seem to index sleep pressure, and they reduce the more you're asleep. But very, very importantly, what Vlad showed is that these sleep intrusion are predicting behavioral errors in a region-specific fashion. So if you recalled, so if you having rats doing a task, so in this case, these rats were asked to reach for a pellet of food. What Vlad showed is that if you recalled sleep intrusion in regions that are involved in the task, here the frontal region, you will see that rats are more likely to miss the pellet of food when you have more of this sleep-like intrusion. So you can see here, when you have misses, you have more of this sleep-like intrusion over frontal areas. You don't see this relationship between parietal areas that are not involved in this task. So it seems that when you're getting tired, you will see more and more of this sleep-like rhythm in the brain. And if they occur in brain regions that are involved in a specific task, then they can be associated with mistakes and errors in that task. So we try to replicate these findings, again, with using intracranial recordings in humans. So we were interested in psychomotor vision tasks. So participants had to respond to visual stimuli as fast as they could. So obviously when people are getting tired, so after sleep deprivation, so the bottom graph, they make more slower responses. They are not paying so much attention, so sometimes they become very slow. So we were interested in this work to differentiate between fast and slow responses. What's the difference between you being fast and you being slow? And what we found is that very slow responses, so what's called behavioral lapse, is accompanied by what we call a neuronal lapse. So when you're yourself behaviorally becoming slower, it can be because your neurons themselves are becoming slower and they have a longer latency to respond. So already at the response survey, being tired is not only responding slowly, but also seeing slowly. So the visual response themselves are being slowed down upon sleep deprivation. And this is associated by an increase in slow frequency power in the same region. So again, something that is reminiscent of sleep. This work has been developed by Bessie Hung, Giulio Bernardi, Simone Sarasso in non-invasive recording, so scalp recording, where they showed that similarly you can observe this increase in slow sleep-like frequencies as people are getting more and more tired. And very importantly, these sleep intrusions are predictive of behavior. So you can predict the behavior and behavioral errors in specific tasks by recording this type of slow sleep intrusion in networks that are involved in this task. So building on this previous work, what I tried to see if whether sleep intrusion could occur outside acute sleep deprivation. So to see this sleep intrusion, do you have to push the brain to its limits to have people not sleeping for a long period of time? Or is it something that can happen in a normal day in our lives without any sleep deprivation? So for example, you right now. So that's something I studied. We published recently with Nao Suchiya, Jenny Wins, and 2 students, Tegan Mackey and Angus Burns. What we did was a design that allows us to look at both the physiology, so especially these markers of sleeps, but also the behavior, to how you interact with your environment, and subjective experience or phenomenology, so what's going on in your brain. So to do that, we use a classical sustained attention to response task. So people have to monitor a screen where we present stimuli, and they have to press a button each time they see a stimuli. So in the digit task, for example, each time they see a digit, they have to press a button. The only instruction they have is they have to withhold their response for the digit number 3. So they have to answer to all digits, but when it's the digit number 3, they have to suppress their response. As you can imagine, they very often press the response button for the digit number 3 because it's a rare event. What we did was to also interrupt them from time to time and ask them whether or not they were paying attention to the task. So whether the task focused, mind blanking, or mind wandering. So that's the proportion of the different mind state during the task. So we replicated a very frequent finding in the mind wandering literature, which is that people pay attention to the task only half of the time, and the rest, they are either thinking about something else. So that's what we call the mind wandering, or thinking about nothing. That's what we call the mind blanking. These different states are also fluctuating during the task. So the more you progress in terms of blocks, the more people will stop paying attention correctly to the task. Obviously, not paying attention to the task has consequences on behavior. For example, people respond less accurately to no-go trials and make more impulsive responses. We interestingly saw differences between these two modes of not paying attention, so mind wandering and mind blanking. Mind wandering was associated to faster responses, so a form of impulsivity, while mind blanking was associated with slower responses, so a form of sluggishness. But both mind wandering and mind blanking were associated with a lower vigilance state. So people had smaller pupil and lower vigilance compared to being on task. So it seemed that although you have a bit of differences between states where you mind wander or state you mind blank, they both correspond to a state of low arousal. So to see if this was related to sleep and sleep intrusion, we detected a marker of sleep in the scalp EEG, which are slow wave. So we detected using the same algorithm that will be used in sleep. We detected slow wave during sleep. And what we first found is that these slow waves were actually correlating well with both an objective marker of arousal, so pupil size, but also a subjective marker of arousal through vigilance ratings. So the more you have this slow wave, the smaller the pupil size were and the less vigilant people felt. So the slow wave seems to be a good marker of subjective fatigue. And what we wanted to know then was whether it was predictive of behavior. So these slow waves are detected at the electrode level. So for each electrode and each trial, you can associate the trial with the presence or absence of a slow wave. And you can use this information to predict the behavior on that very same trial. For example, imagine that I present you with a digit. I want to know whether you're going to respond to that digit depending on whether or not I'm seeing a slow wave in a specific electrode. So when you do this, what you can do is then plot topographies of which electrodes are predictive of behavior based on the presence or absence of slow wave. So that's the plots I'm showing here. And what you can see is that when we observe slow wave in the front of the brain, we're able to predict false alarms. So when people are responding in trials where they were not supposed to respond. But slow wave in the posterior areas were more predictive of misses. So when you failed to respond and you were supposed to respond. So it seemed that there is this difference between slow waves occurring in the frontal part of the brain and the posterior part of the brain. Slow wave occurring in the frontal part of the brain are associated with more impulsivity, so false alarm, and slow wave in the posterior part of the brain, so they do with more sluggishness. And this was reflecting actually through reaction times. Slow wave in the front of the brain predicting faster reaction time, and slow wave in the back of the brain predict slower reaction times. So we seem to have with slow wave a good predictor of your ability to perform, and a predictor that will predict different types of errors depending on which brain region are getting tired. This is an approach we could replicate with the mental states. So this time contrasting when you're thinking about something else versus being on task, or thinking about nothing versus being on task. What we observe is when people are thinking about something else, they have more slow waves in the front. These slow waves have a higher amplitude and a bigger slope. When they are not thinking about anything, we still have more slow wave in the front, but these slow waves tend to be larger in the back of the brain. Evidencing that, again, between front and part of the brain, you might have differences in the impact of slow wave on subjective experience. We replicate that these findings and this frontal-anterior contrast with modeling. Something that was quite interesting and relevant to current topic debates in consciousness was the fact that although we see this divide between front and back of the brain, I don't think our results can really say that when you're switching off the front of the brain, then you might still be conscious because you're mind wandering, but if you switch the back of the brain, you are not. So I don't think we can use this to say, well, back of the brain is more important for consciousness than the front. And this is because these waves are actually traveling. So the wave we see in the back are sometimes actually coming from the front. And it's possible that the state of mind blanking is a deeper state of local sleep in a way, with waves that have the tendency to be present both in the front and the back of the brain. We can discuss more of this later in the discussion part, but I think it's important to remember that this wave potentially travel. So this is a bit of the state of well, the summary for these findings here, that we have the ability to detect this sleep intrusion in awake participants, even if they are not sleep deprived. So these participants were really you and me going about a normal day. They still show sleep intrusion when they're awake. And these sleep intrusion have consequences on both their behavior and their subjective experience. And the consequence of this wave really depends on where they occur within the brain. So one single mechanism could have different cognitive consequences depending on the brain areas impacted. So very, very briefly, because we're already at the hour, we just mentioned new directions I'm taking with this work. One is to try to explain attentional deficits. So I'm especially interested in ADHD, because ADHD is often depicted as a dysregulation of arousal. So the reason why people have a deficit of attention is because they have a harder time to manage an optimal level of arousal. And this dysregulation of arousal could come from actually sleep disturbances. So ADHD adults and children have a very high prevalence of sleep disturbances compared to the general population. So what we want to study here is whether we can explain ADHD symptoms by potentially the occurrence of sleep intrusion. So a non-ADHD individual will be sleeping well at night, so in a global way, and will be well awake during the day. And an ADHD individual will potentially have a disturbed sleep with more intrusion of wake within sleep, and that will translate into more sleep intrusion during the day. So we have primary studies confirming these insights using two different sustained attention tasks. When we record the EEG in ADHD compared to control, whether ADHD were on and off the medication, we saw more slow waves in the ADHD group compared to the control. So it seemed to have more sleep intrusion than non-ADHD individual. And this seems to be correlated with the attentional deficit as behaviorally measured. We're also trying to manipulate this amount of sleep intrusion using pharmacology. One thing interesting for this link between sleep and attention is that they're regulated by the same neuromodulatory system. In particular, noradrenaline, dopamine, serotonin are involved in both attention and arousal. So we try to manipulate this neuromodulatory system using drugs in healthy participants and see what the impact on behavior and electrophysiology. And what we found in particular is if you increase the level of serotonin, which usually has a sedative effect, what we can see is an increase in this sleep intrusion compared to a placebo. If you try to boost vigilance, on the contrary, you don't find anything. So it's really when you lower vigilance, you start seeing this sleep intrusion within the brain, and these sleep intrusion are again predictive of performance in a task in a region-specific way. So if slow waves are more prevalent in the back of the brain, you observe a slowing down of reaction times, more misses, and if they occur in the front of the brain, you have actually faster response. So moving forward, we're interested in studying this sleep intrusion in a more varied clinical population, seeing if it's predictive of aging. With after a PhD student working with me, I think who is present here, we're studying in trying to see if we can generalize the concept of local sleep to other markers and slow waves, see if they're associated to function and positive aspects, such things like creativity, and whether we can non-pharmacologically manipulate sleep intrusion. So thanks a lot for the work. I want to, thank all the people that have been working with in the past 10 years. It's an important network of international collaborators, but most of them are also friends. So I'm really, really thankful to them, and thankful to my new team and institution for pursuing this work.